So let me explain how things are going to be going for the next 2 weeks, basically. So we're STAAR testing already. We're getting ready for the STAAR test. Sorry, we're reviewing. We're not STAAR testing. We're reviewing. Everyone was supposed to take the test yesterday. Remember, I told you to pretend like it was a real test. I asked you to use no notes, no phones, no nothing, because I want that data to help me determine what notes we need to focus on and what notes we don't need to focus on. So not everyone got to finish the test, so I'm going to wait one more day before I collect data. That being said, today I'm going to go over everything because, again, I haven't collected the data to see who needs to go over cells and who doesn't. So today we're just going to pretend like no one knows anything about cells, and we're going to cover everything. So it was requested by my 8th period class to do Blue Kit. I hope you all know what that is because they loved it. So I made this. We're going to play it for 10 minutes to review cells and viruses. All right. Good job, guys. Good job, everybody. Oh. Okay, so from this, hopefully, if you got all the questions right, then this is a topic that you're very, very knowledgeable in, and you shouldn't have to focus on studying these notes and these terms. Like I said, every day I'm going to give you a new topic. If there is a topic that you don't know that you're doing really bad on these games on, then I do highly encourage you to focus on this when you're studying for the biology STAAR test. All right, so again, I have your new daily work folder right here with your STAAR review. Today we're going to start working on the graphic organizer. I'll tell you which slides you're going to be doing, but that's going to be your homework assignment, and that's like a study guide for you. If you need help or if you don't understand something, let me know and I'll tell you the answers because I want you to have the right answers to study. So this is the actual STAAR review with all of the answers, their terms, their definitions, their things for you to review. So if you did bad in today's game, click on this, read those terms, look at those definitions and those concepts. That's what you need to focus on for today. This is the biology STAAR 10-day review. So for example, if we all knew cells, then I would skip today's lecture and we would go on to the next thing. But like I said, since today I'm just going to pretend like we don't know cells, I'm going to show you how we're going to be doing things. This is the actual slideshow zoomed in. If you're looking on your own, I'm going to start having hyperlinks so you can just click on the day and it'll take you to those sets of notes. Again, later on when we get closer to the STAAR, if you forgot something and you want to go back, just click on what you want to go to and it'll take you right to it. So today is the 20th and we're going to be doing cells and viruses. So again, cells are the smallest unit of living things. Simple cells are prokaryotic, they lack a nucleus. Complex cells are eukaryotic, they do have a nucleus. These are the organelles, and the primary function for those organelles are right here. Cell membrane surrounds the cell. Cytoplasm is kind of like that gel-like stuff in the middle. The nucleus holds the DNA. Mitochondria gives you energy. Ribosomes make protein. The ER moves things. The lysosomes have enzymes. The vacuoles hold things. Cell walls only exist for plant cells. They provide structure. Chloroplasts also only exist in plant cells. They perform photosynthesis. Homeostasis describes equilibrium. Remember, the body wants to be balanced, and on a smaller scale, the cell wants to be balanced. It wants the perfect amount of salt, perfect amount of potassium, perfect amount of calcium, all of those other micromolecules. So how would the cell maintain homeostasis in a salty hypertonic environment? How would the cell be responsible for maintaining homeostasis? And we're going to talk about those right now because that's molecular transport. So I heard someone say they forgot about transport. So this is a topic you would have to go in yourself, review it. look up those terms once more. Homeostasis is achieved by letting materials go through the membrane. Remember, active transport needs energy because it goes against the gradient. It goes from low to high. Passive transport does not need energy because it goes with the gradient. It goes high to low. We're not doing this. So now we're comparing viruses and cells. Viruses are not living because they don't have the 8 characteristics of life. They can't make their own energy. They can't reproduce on their own. They need a host. And they can cause diseases. Cells like bacteria are living. They do make their own energy, or they eat to get energy, and they can reproduce on their own. And these also cause infections. We're not doing this right now. Talking about HIV, how it's a virus. It cannot be treated with antibiotics because remember, viruses are not living. And then once you're done with the notes, you can click here to return to the table of contents. It'll take you back, and then you can click on another day to review those notes. So that's how the notes work. You have that right here. I'm obviously not done fixing it. So next week, it'll be for next week's topics, but right now I only have today, tomorrow, and Thursday. Again, this folder, purple folder that says warning, has a lot of material, So if you were absolutely clueless about today's topics, I highly encourage you to go into this folder to read those notes over cells, organelles, transport, and viruses. We're in week 5. Here on Tuesday, we played the warm-up game for 10 minutes. We're going to do the notes. Oh, I'm sorry. We already did the notes. We're going to do the STAAR assignment number 1, and we're going to go over the extra resources. And then your homework are going to complete slides 1 through 8 in that graphic organizer. So clicking on Tuesday's folder, again, here are the topics once more. So if you didn't understand at all the Blue Kit game, come here, read these topics, and hopefully you'll be able to catch up. You don't need to do the attendance check if you're in Zoom or if you're in class because it's just one question it asks if you're here. You don't need to play the game again unless you want to. And right now we're going to do the assignments, cells, and viruses. So this is going to be for a grade. Every day it's going to be for a grade. You're going to have one every day, and I'm going to go over them with you every single day. So this would be a perfect time to open it up so that way we can answer the questions together. The whole point of this is I'm going to teach you how to focus on certain words because, of course, the STAAR test is going to give you paragraphs most of the time, or it's going to give you tables with a bunch of information, which really you don't really need. If you pay attention to those keywords, you can answer the question very quickly. So that's what we're going to do right now. I printed it out so I can write on it. All right, so I don't think the questions are in mixed order. I think they're all the same. So number 1, we all have the same number 1. Both Eugenia and cyanobacteria are photosynthetic unicellular organisms found in pond water. So I'm going to highlight those keywords. We have Eugenia, which is a protista, in case you forgot. We have a cyanobacteria, which it says bacteria, so that's a bacteria. They're photosynthetic, meaning they use photosynthesis. They're unicellular, meaning it's one cell, and they're found in pond water, so they live in the water. So we want a feature that distinguishes them from each other. What's going to tell them apart? Well, cyanobacteria, all bacterias are prokaryotic. And if you remember, pro means no. Remember, prokaryotic cells have no nucleus. Their DNA just floats around. Eugenia is a protista, and that is a eukaryotic cell. And if you're a euc, you have a nuke, meaning if you're eukaryotic, you have a nucleus, surrounding your DNA. Oh yeah, it's gonna get cheesy, guys. Real cheesy. Yeah, exactly. That's the only way I can remember it. If you're a uke, you have a nuke. So that's the difference between the two. And if you're looking at your answer choices, right here, presence of a nuclear membrane. Ukes have a nukes, pro means no. Another thing you can look at are the other words. Maintaining homeostasis, ability to reproduce, presence of ribosomes. All cells have ribosomes. That's part of the RCDC. Everything that's alive has ribosomes, has cytoplasm, has DNA, and has a cell membrane. So everything has that that's alive. Being able to reproduce and homeostasis, those are the 8 characteristics of life. So every living thing has that. So all 3 of these exist in every living thing. They wouldn't be used to tell the difference. Number 2, hopefully this question looks familiar to you. Some students use information they gathered from the lab investigations to prepare a table. So we have cell 1 and we have cell 2, and the question's asking which of these correctly identifies the 2 cells described in the table. Well, cell 1 is smaller than 5 micrometers. Cell 2 is larger than 10. Cell 1 does not have a nucleus. Cell 2 does have a membrane-bound nucleus. Cell 1 does not have membrane-bound organelles. Cell 2 does have organelles. Cell 1 has circular DNA. Cell 2 has linear DNA. And hopefully the second one is able to tell you the difference between these cells. No nucleus in cell 1, nucleus in cell 2. So that means cell 1 is prokaryotic and cell 2 is eukaryotic. Again, pro means no nucleus. If you're a euc, you have a nuke. All right, the next question shows a student using a microscope to study a prepared slide of a single-celled organism. So keyword, single-celled, meaning it's one cell. A single-celled organism can be classified as a prokaryotic based on the absence of, well, what is the one thing that a prokaryotic is missing? The main thing, it's missing a lot of things. But remember, pro means no nucleus. So the thing it's missing is a nucleus. I want to point out again, the other organelles are a ribosome, a cell membrane, and a chromosome, which is DNA. Those 3 belong to the RCDC. Every living cell has these 4 characteristics. Again, R stands for ribosome, C stands for cell membrane, D stands for DNA, and the other C would be cytoplasm. All living cells have those, so it wouldn't be able to tell you the difference. Next question. Students use a microscope to look for structures present in 4 different cells. The students placed an X for each structure that was viewed for each cell on the table shown. So we got cell membrane, cell wall, chloroplast, DNA, and a nucleus. The question is asking which cell that was viewed is most likely a prokaryote. Again, when you're dealing with prokaryotes, pro means no nucleus. So you don't need to look at all the other structures. All you need to focus on is which one does not have a nucleus. And that would be cell X. So that one would be automatically the prokaryote. All the other cells would have to be eukaryotic. All right, so now we have different types of cells. We need to know which table headings we should use for the 2 groups. So this deals with the structure of cells. Hopefully you can visually see the difference between these 2. Here the DNA is floating around. Pro means no nucleus, so this is a prokaryotic cell like bacteria. Here we have a nucleus, and if you're a euc, you have a nuke. If you want to get more specific, this would be a plant cell, this would be an animal cell. So when you're looking at these, the answer choice should be A, prokaryotic cell for the first one, eukaryotic cells for the second one. These deal with mobile cells and motile cells, fungal cells, bacterial, animals, and plants. Just looking at those, you'd be able to cross out those options because, well, they're just not making sense. If you have questions about that, let me know and I'll go over it with more detail. All right, number 6. Influenza flu has 4 types of viruses, A, B, C, D. Influenza A and B are the most common human types seen in the winter season. Symptoms may include fever, cough, fatigue, body aches, etc., and typically occur 2 to 7 days after infection by the virus. Influenza is more serious in elderly patients. The information suggests that the reproductive cycle of influenza is. So on this question, there's a lot of reading going on, and that's what the STAAR test is going to do. Sometimes it's going to give you questions with a lot of reading material. We're looking at the reproductive cycle only. That's what we care about. Obviously, we know that this is a virus. It's the flu. And one of the key sentences or key parts here was that symptoms typically occur 2 to 7 days after infection. So we do have 2 types of reproduction for viruses. Based on the reading of the choices, we could tell it's lytic and lysogenic. So those are some of the words you're going to have to remember the difference between those 2. Which one's lytic, which one's lysogenic? Lytic happens more fast. I'm sorry, it happens faster than lysogenic. Lytic's going to happen quick. You're going to see the results so fast, kind of like chickenpox. Lysogenic's going to happen slow. It's going to take a longer time to see that progression, kind of like HIV. So based on this, if you can typically see symptoms 2 to 7 days, we say that's fast. Didn't happen over a lifetime. It happened 2 to 7 days. So we know it's faster. We know it's going to be lytic. And then you're just reading which one of these correctly matches lytic. B, because of the quick onset symptoms of after infection, or D, because the virus causes respiratory illness. Respiratory illness does not deal with reproductive cycles. So that would have to be B, because it happens faster. Question 7, the picture shows a 3D model of a virus called a bacteriophage. Bacteriophages can infect bacteria such as E. coli. So we've talked about viruses before, and we've talked about bacteriophages. Bacteriophages eat or attack bacteria. In case you forgot, that's one of the terms. So in what way are bacteriophage and E. coli alike? So bacteria is a prokaryotic. Pro means no, nucleus. E. coli is a bacteria. Oh, just kidding. I got that confused. Sorry, it's a virus. E. coli is a bacteria, and pro means no, nucleus. So the difference between those 2, one's alive, one's not. Reading the answer choices, how are they alike? How are they similar? What makes them kind of the same? They do not have identical genomes. They do not contain antibodies. They both do lack membrane-bound organelles. Reproduced by mitosis, that only exists for living things, not non-living things. So that does not exist for a virus. So the only answer choice would be B. All right, number 8. Viruses can be transmitted through air, water, food, insect bites, and direct skin contact. Once a virus gains entry to the body, it invades a host cell in order to blank. So what's the whole point of a virus invading a host cell? Metabolize host proteins and grow. Viruses do not grow. Synthesize antibodies for defense. Viruses do not have antibodies. Deactivate the host cell's defense. It doesn't try to deactivate the host cell's defense. It kind of slows it down by attacking, but it doesn't deactivate it. Access for cellular respiration for replication. That's the correct answer because remember, it needs a host cell for replication. All right, number 9. Which of these describes the difference between a virus and a cell? Well, the most common thing before we even start reading the answer choices, viruses are not living. Cells are living. So reading the answer choices, cells reproduce independently and viruses require a host. That's true. Viruses do need a host. Viruses have membranes made of proteins and cells have membranes made of nucleic acids. That's not true. Cells' membranes are made of lipids. Cells contain protein and viruses contain only carbohydrates. Viruses do not have carbohydrates. They only have proteins and DNA. Viruses have flagella and cells have only cilia. Cells have both. They don't only have that. So the only choice can be A. Going to question 10. A student produces a label drawing of a virus for a presentation. The student states that the capsid has a function similar to nuclear membrane found in animal cells. Which of these describes the similar functions of capsids and nuclear membranes? So we're looking at the similar functions of a capsid and a nuclear membrane. So when you're looking at these, here's the capsid and it's surrounding the DNA. Nuclear membranes surround DNA as well. It's not a cell membrane, it's a nuclear membrane. So when you're looking at the similar functions, both code for proteins, that's not what they're doing. They're holding DNA. Both protect genetic information. That can be true. Both provide energy. Well, it doesn't say anything about energy. And both transport proteins throughout the structures. Doesn't say anything about transport. So that would be C. They hold the genetic information. All right, number 11 and 12 deal with cell transport. Membrane transport. Number 11, use the following information to answer the next 2 questions. Oh, that should say 1. That's a typo. In the picture below, the cellophane bag models a selective permeable cell membrane. The bag contains a solution of 98% water and 2% salt. If the bag is placed into beaker A with 100% water, what type of solution is found in the beaker compared to the bag? All right, so this is dealing with osmosis. That's one of the terms you're going to have to remember. And water goes from high to low. And we have 3 different types of osmosis. We have hypertonic, which shrinks. We have hypotonic, which gets fat like a hippo. And then we have isotonic, which stays the same. So if we're putting the bag into beaker A, we have 98% water in the bag. We have 100% water in the beaker. Water's going to go from high to low. So water is going to go from the beaker into the bag. And if water is going into the bag, water is going to get fat. The bag's going to get fat. So that means the bag is in a hypotonic solution because it's getting fat. Last question, number 12. The diagram shows cellular activity across the cell membrane. Which two processes does this diagram most directly model? So the whole point of moving across a cell membrane, this does deal with transport. And if you remember, the whole reason why we need to move molecules is to keep a balance. And, whoops, I spelled balance wrong. The word for keeping balance is homeostasis. So that's one of the processes that it models. Energy conversion, we're not dealing with energy conversion. Synthesis of new molecules, we don't make anything new here. Energy conversions, again, we're not converting energy. We're dealing with homeostasis, yes. And we're dealing with transport, we're moving things, yes. So the correct answer would be D. So we're gonna be going over more of these questions later. Ooh, almost done. But that's it for today. So again, if you didn't understand these, you need to go back, you need to review that material. So now for your homework. Oh, sorry, I forgot to tell you. If you click on these topics, it takes you to more games to play and to review. Not mandatory, again, it's just practice. If you click on them, it'll take you here. Watch videos, play games, study, review. But for Tuesday, I want you to complete slides 1 through 8 of your graphic organizer. So on your STAAR review, the first thing is your graphic organizer. It's kind of like your review template. You don't have to print it, just typing everything. You're going to do slides 1 through 8, fill in the blanks, answer the questions. If you need help, let me know. Sorry that took a long time. Every day is going to take a long time because we're going to be talking about every topic. If you have any questions, stay on. If not, you're free to go do your graphic organizer.